Hi there, everyone. We are so happy to have you, whether you're joining us here today or will be watching later on the Cure GABA-A YouTube channel. My name is Monica Joanna Alancave, and I'm the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential therapies for GABA-A variants by partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trials. Today we welcome Cordelia. Cordelia, who is a fifth-year medical student from Copenhagen, Denmark, currently pursuing her master's thesis in personalized medicine in genetic epilepsy at Stanford University's Juliet Knowles Lab and Philadelphia Epilepsy Hospital. Her career goal is to integrate research findings into patient care, particularly for children lacking treatment options. Supervised by Dr. Juliet Knowles and Professor Reike Moeller, her project aims to establish a framework linking genetic diagnosis to precise treatments for pediatric epilepsy. The study addresses challenges in translating genetic data into clinical applications and investigates genotype and phenotype correlations in GABRB3 related epilepsy to advance precision therapies. I'm handing it off to you. Please help us understand genotype-phenotype correlations and precision treatment.
Hi, everyone. Thank you so much for listening to this meeting and this presentation. I'm really excited to be able to share some research that I'll be doing for the next year. My name is Cordelia Smith, and I'm a fifth-year medical student at Copenhagen University. My project is about the beta-3 part of the GABA-A channel, and I'm going to be looking at loss of function patients. I'm working together with Stanford University, the Philadelphia Epilepsy Center in Denmark, and also Copenhagen University. I'm going to start by just giving you an overview of this presentation. First, I'm going to do some background information about the GABA-A channel. You might know some of these things already, but I'm thinking it's kind of a good idea to ease you into the project and what it's really all about. Then I'm going to be talking about this project in specific, and I'm going to be talking about the methods that we'll be applying, as well as how we're hoping to create some really meaningful results for patients. Lastly, I'm going to just let you know what kind of future directions we're seeing in terms of science and where we would like to go from here. Yeah, so first off, the GABA-A channel, as you might know, is a channel that's located all throughout the central nervous system. It consists of 5 subunits, so that's the picture I have up here on the right. It's kind of like 5 subunits in a circle. Most commonly, the combination of those subunits are 2 alpha subunits, 2 beta subunits, and 1 gamma subunit. There are other types of combinations as well. This is just the most common one. And so I'm going to be looking at one type of a beta subunit called the beta-3 subunit. The GABA channel, this channel, is a ligand binding channel, which means that as soon as GABA or something like a benzodiazepine medical ligand will bind, it opens up, and that allows negatively charged ions to flux into the cell. And when these negatively charged ions flux into the cell, that's what causes the cell to be less prone to create a firing. Or in other words, that's what decreases the risk of seizures. It's generally called decreasing the excitability of the cell. Generally speaking, the GABA is an inhibitory channel, whereas the NMDA receptor and other very widespread receptor in the central nervous system in the brain, that's the excitatory receptor. So when GABA is mutated, that's what causes kind of an imbalance and means that the NMDA part has more power and the brain becomes susceptible to seizures. Then down here in the right corner, you can see I have a circle with a picture of a 2D structure of the beta-3 subunit. And each little dot is a variant location that's possible. So when we're talking about these mutations in the subunits, it's not just one mutation in one subunit, it's actually many different types of variants within that mutation. And that's what makes this so complex and what creates this a difficult puzzle to solve for many scientists. In my project, we're going to be looking at many different variants for the GABA-B3 subunit and try and make sense of them. And then we're going to be looking at one specific variant using a mouse model. I'm going to be talking more about that later. Then this part is maybe something you're more familiar with. Generally speaking, we're kind of dividing it into epilepsy on one hand, and then other outcomes of the mutations on the other hand. As I told you before, mutations in the GABA-A usually cause a more excitable brain, meaning we have more seizures. And these seizures can be really damaging to a brain, especially a young brain. And sometimes that's what causes the other outcomes. Things like delayed or absent language and motor skills, intellectual disability ranging from mild to very severe, and also neuropsychiatric disorders in many, many patients. In our cohort, it's about 61% who have some type of neuropsychiatric disorder. Sometimes it's the mutation in itself that causes some pathways in the brain that we don't understand yet to be activated or downregulated, and that's what's causing these other outcomes. And that brings me on to an important part of this presentation, because this so complex diagnosis requires personalized medicine. But personalized medicine isn't just easy to get to. There are quite a lot of challenges posed to researchers at the moment. One of them is that these variants have specific phenotypes for each variant, and this is where you really come in. And I just heard from Monica that every single patient who signed up via the Cure GABA-A community, they're automatically linked to Matrix and other types of places where they can load on their patient data, and then the researchers can get access to that. And that's so valuable to us. That means that when you sign up through the Cure GABA-A, you're really helping us understand the phenotype, how the children are doing, and how that relates to the genotype. So that's the variant they have. It's so great information for us, and it's going to help us make sense of what's going on with these genes. So really important. Another important point is that when you sign up through the Cure GABA-A, you're also automatically linked up to some of my amazing supervisors that I'm going to be mentioning in a minute. One of them is Rikke Müller and another is Sebastian. And they're both part of this personalized medicine for genetic epilepsy project and are experts in the field. So that's one part where you can really make a difference. Another challenge is that these genetic diseases are so rare that sometimes it's hard to find one trend that's actually strong between patients. I think that a way of solving that is to work together internationally and to have strong international collaborations where we can share all of our data so that we get more data on these rare diseases. Another future and very promising solution is computer models, and they're going to be helpful to predict and create prognosis for each variant. Then the third challenge is that we have a lack of guidelines for physicians. So a lot of physicians have patients that have a genetic diagnosis, but they're not used to handling it, and they don't actually know what to do, even though they have the diagnosis. And I think it's a responsibility for researchers to not only do the research, but also translate that into something that's useful in the clinic. We're talking a lot about simple decision trees for clinicians so that they can either see symptoms in their patients or know something about the variant, and then from there make the right decisions to treat in the right way. One last point that's really important and very challenging is the biological complexity related to these genetic diseases. One mutation and one variant can go off in one direction, whereas another can go off in a completely different direction. And understanding how these connect and impact each other, that's a big chunk of a challenge that we're facing. It's also part of what we're trying to solve in this project in particular. We're going to do a protein pathway network analysis to try and make sense of that. So that leads me on to this project. This project is called Genotype-phenotype Correlation and Precision Treatment for Epilepsy Related to the GABA-A beta-3 Loss of Function. There's me, I'm at the top, Cordelia Smith, and then there are my two amazing supervisors, Rikke Müller, a professor at the Philadelphia Epilepsy Center in Denmark, who is a professor in genetic epilepsy, and she's also one of the experts you'll be linked up to when you sign through the Cure GABA-A. And then Dr. Juliet Knowles, who's actually one of the recommended physicians through Cure GABA-A. She is located in California at Stanford. And at Juliet's lab, I'm going to be working for the next year to look at her mouse model of this genetic epilepsy. She's amazing, and she's also a clinician treating patients with rare diseases. Yeah, so the way we're looking at this is that there's a problem. The problem, as I mentioned before, is that many children receive genetic diagnoses, but there's not really enough knowledge to know how that should lead to a prognosis, or if there are any options of targeted treatments, and usually these options that might be available are very slim. Our approach to this is that we want to combine mouse data, so that's going to tell us a lot about the biology and what's going on in the brain, and then we want to combine that with the people data, which is from the cohort, so looking at how are the patients actually doing, and then we're hoping that these two will kind of overlap, and that's going to create a whole new picture that's a little bit clearer, and that's going to point us in the right direction for targeted treatment options. So put more plainly, these are the aims of our project. We're going to, perform a detailed genotype-phenotype correlation of individuals with loss of function GABA-beta-3 related epilepsy. And then we're going to, two, use single-cell transcriptomics to define novel precision therapeutic targets in heterozygous mouse model called GABA-beta-3 D120N. So the D120N part, that's the variant part. Which is a mouse model of GABA-A beta-3 related early onset epilepsy. Then I'm going to be talking about the methods that we'll be applying, just to let you know exactly how we're going about doing this. So the first part, the genotype-phenotype characterization, that's actually just gathering patient data. This was the part where you come in. And right now we have about 70 patients that are registered or located at the Philadelphia Epilepsy Center in Denmark. And from them, we get a lot of information about the age of the seizure onset, the language, motor skills. Some of them even have EEG patterns available. Those are very valuable to us. It lets us know exactly what type of seizures patients are experiencing, and that lets us know more about what's going on in the brain. Then, as I mentioned before, the neuropsychiatric disorders are common in many patients. And lastly, really importantly for this project, we're going to be looking at what treatments are working, which ones aren't, and some of them are even worsening the seizures of the patients. So a really important point is the treatment response in patients. The genotype, which I have listed here on the left side, that's more about where is the variant located. So if you remember the 2D structure I showed you before, it's kind of like which point is it at, and does that matter? Is it altering the channel structure? Really important parts. Then that brings me on to this slide, which is showing what I'm going to do for the next year at Stanford in the US. It's a mouse model available at the Juliet Knowles lab. And in this mouse model, we're going to do transcriptomics. These transcriptomics are going to let us know which protein pathways are up or down regulated. So basically, it's going to tell us when there is this mutation, this is what's going on in the brain. And that's really valuable because that lets us match it to a large drug database that's available at MIT. So that's what I have down here in this corner. It's called root analysis, the method we're applying. It basically lets us match about 6,000 FDA-approved drugs with the genetic data from the mouse, and this data about protein pathways up and down regulated. And that's why we can make what's called a connectivity map. That's what I have down here in the right corner. So you can see that I have the drug name right in the left column, and that's the information from the drug database. Then I have the drug description in the middle. that tells us how it's working and which pathways it's targeting. And then out here, I have the genes that are connected to that. And these genes are not genes that we have to look at individually. They're kind of genes that are impacted by this mother gene that we're looking at. So in this case, it's the GABA-B3 loss of function mutation. So when we have this mutation in the GABA-B3, that impacts all these other genes that we're going to be able to tell if are up or down regulated, doing something that's called a differential gene analysis. That's really exciting. It's going to show us how we're going to connect the dots. And that leads me on to this slide. This is what I call meaningful results, because we're applying our data from the mouse and from the people, so that's our cohort. We're going to hopefully find some drug database matches, and that will allow us to repurpose drugs that we already have, which is very different from having to develop new drugs all the way from the beginning. That's going to take much longer time. We're hoping that this project can lead to a repurposing of drugs, and then that way bring results to patients much faster. And then lastly, I just want to tell you about future directions within science and how this project can kind of lead to new ways of approaching rare genetic epilepsies. This template that we're applying, as my supervisor from Stanford calls it, it's kind of like a new template for understanding rare genetic diseases. We're applying both mouse data and people data, and we're bringing it together. So that could be a new approach that could be copy-pasted onto some other rare genetic epilepsies. Then we have something that would be really, really great is to create some clinical recommendations based on variants. So saying if you have this variant in the gene, we recommend that you go down this route and you do this for your patients. That would be amazing. And then we have, of course, the drug matches that we're hoping for. One thing too is that we're going to try and, of course, see if they can be repurposed in some way and brought to an off-label use. Another direction that's really important is that we're going to take those matches back into the lab, and we're going to see how they're going to work on the mouse and try and understand exactly what's going on in the brain. That's going to be so important. That was my last slide, and then I just want to say thank you for listening. And if you have any questions or interest in being part of this GABA-B3 loss of function cohort, you are, as I said before, if you sign up through the Cure GABA-A community, you'll automatically be linked to my amazing supervisors at the Philadelphia Center. So that's Sebastian Oitz and Regge Müller. But I also just wanted to put in my personal contact information if you want to get in touch with me directly. I'll see you in about a year, and I hope to bring some really great results with me. Thank you.